Okay, I'm very excited to introduce our next speaker, joining us for the first time at RAADfest, albeit remotely. He is the Wyss Institute Professor of Genetics at Harvard Medical School. Dr. George Church leads synthetic biology at the Wyss Institute, where he oversees the directed evolution of molecules, polymers, and whole genomes to create new tools with applications in regenerative medicine and bioproduction of chemicals. He developed the first direct genomic sequencing method and helped initiate the Human Genome Project and the Personal Genome Project. Please welcome Dr. George Church.
Yeah. Well, hi, it's great to be part of this event. Thank you very much for tolerating my virtual nature. This, hopefully you're seeing one slide here, is the thank you, beginning of many thank yous that I'll be making. This is thanking some of the granting agencies at Harvard and MIT in particular that have provided wonderful students and postdocs, which I will mention as we go along. I'm talking about technology to radically alter our genomes. You heard a little of this in the panel that we just had, but I want to explain why and how it has to be, radical and how we go about that. So there'll be plenty of time at the end for Q&A. I may even finish a little bit early so that we can do that. So part of the reason we're having this discussion about radically changing a genome is that we have undergone this exponential change. We live in an exponential technological time in many ways, not just computers, but all sorts of biotechnologies, including, in this case, reading and writing DNA in red and blue color code here, where importantly, the y-axis is changing by factors of 10 per increment. So this is a very rapidly, maybe deceptively rapidly changing over a linear time frame on the x-axis. And what's happened is we've been following sometimes as slow as the incredibly fast Moore's law for computers, and sometimes faster. And in particular, we've improved about 20 million-fold our sequencing costs, maybe 5 factors of 10 in quality, and similar for writing DNA, raw DNA. But what about therapeutics? That's what this is about next. Isn't therapeutics going up in cost rather than exponentially down in cost? And here's two of my companies. I'll keep thanking them, various ones along the way. 64X and Helix Nano. But I'm gonna say the therapeutics can come down radically, as Liz just alluded to, but I'm gonna spell out quite clearly here. The cost for rare diseases is in the $1 million to $3 million a dose range. And I don't know of any exceptions. But if you look at the way that these gene therapies are formulated, they're typically either a viral capsid protein around a double-stranded DNA or lipid nanoparticle around single-stranded messenger RNA. And this very same manufacturing process and packaging and so forth is exactly how the COVID-19 vaccines were prepared. So the economics should be very similar, and it shows the economies of scale. So here you have somewhere between $2 and $25 per dose, in contrast to $3.5 million per dose for the latest gene therapy for rare diseases. And I would argue that's almost all entirely economies of scale. Here you have over 6 billion people worldwide have taken these vaccines that are formulated as gene therapies, and hence we've averted some of the deaths, we still had 7 million excess deaths because of this terrible pandemic. So there are other diseases, health issues that are a similar scale. They're not that many, but pandemics and age-related diseases. 90% of us will probably die of age-related diseases if we don't do something about it. And that's been only, unfortunately, 7 million have died of the pandemic. So why focus on gene and cell therapies? Now we can think of small molecules as the most historically pharmaceutical approach, but they are challenged in discriminating members of a family. So almost all these are targeted for proteins that are members of and some of them are members of gene families, where there'll be many proteins that are related to each other, but they have different tissues they're expressed in, slightly different regulation. And then there's things called splice isoforms, where where it's the same gene, but it's many variations on it, the way it's spliced together at the RNA level. In small molecules, the active sites look all alike for these within a family, within a splice isoform family. But for gene therapies and cell therapies, you can be very specific in saying, I'm interested in this family member and this splice form, and so it doesn't have that problem. Directly connected to the way we do basic biology. And cell therapies have additional advantages. They're a little bit smarter than gene therapies. You can think of it like modern computers have terabytes of information, and the computers I started with had a few bits, tens of to thousands of bits. Now it's billions, trillions. But it's also smarter in that it can extravasate, which is just a fancy term for going across your blood vessels. It can chemotax. It can follow gradients of active chemicals. Okay, so what can we do to make gene and cell therapies better? Well, one thing that we have now is a breakthrough in machine learning, large language models for the experts in the group. But that's not all. It's coupled with multiplex libraries. These are things where we can make, by libraries, I'm talking about molecular libraries, in this case, proteins and nucleic acid libraries, and we can make millions of them. And in fact, here's an example where we made millions of different capsids to improve the delivery, to make it so it evades the immune system. You want to be able to reuse that package, the viral delivery, like the ones that I described for vaccines. They can also be used for curing diseases. You want to evade the immune system, and you want to target it to a particular tissue. If you're lazy about it, it'll just go to the liver. But if you work on it, as you'll see in the next slide, you can really exceptionally target a new tissue and cover that new tissue very uniformly. Or you can target all the cells in the body, as you might want to do for age-related diseases, or all the stem cells, the ones that are dividing. Anyway, that targeting is done by cycles of machine learning called DNA synthesis, where we don't make random changes. We make the ones that are defined by the machine learning. And then we can inject even an expensive animal model like a non-human primate and see where the virus particles go. Now remember, the gene therapies don't contain viral DNA. They contain your favorite gene or genes. This was thanks to Pierce and Eric, published these two papers, and they co-founded Dino Therapeutics, which is all about using this machine learning plus mega libraries. So I'm going to walk through some of the data there to show how it works. This is very simple heat map, meaning color coding, where red means it works better than the original, and blue means that it's targeting away, it's avoiding those tissues. And here's 6 tissues. We did, dozens of tissues from all over the body. And you can see here, red are variations on the virus, where made every possible substitution, systematically changing every position in the viral capsid. And we found places that are better for blood or heart or liver, so forth. And here's an example. One of the first ones that's going out in production is called BCAP1 for brain-directed capsid 1, where we improved over the previous best here in magenta. It now delivers much more efficiency, much more highly specific for the brain, and uniform throughout the brain. So that's BCAP1, again, thanks to Eric and Pierce and their colleagues. And the final slide in this series is showing how we can, that was just changing systematically every possible position in the viral capsid. It's a long protein polymer that folds up into, makes a delivery vehicle. But what if we wanted to change them a lot at once, rather than one at a time for every, systematically, changing 28 at once? And so we took a key 28 on the surface in a key part of the capsid and ask what we can do. And all of our previous work culminated in maybe getting 4 out of 28 changed, and it breaks. It's basically right on the edge of breaking at 4. You go up to 5 or 6, and it's dead. All of them are dead. There's no library formation for it. But with using machine learning, in particular logistic regression, we can get 26, 27, and even 28 out of 28. We can completely change all amino acids and have multiple solutions, not just one solution, but a whole library of solutions, up to 80% libraries where the machine learning has predicted 80% that they would be viable with new functions and so forth. And this is not restricted just to the viral capsids, as I'll show in the next slide. We can apply this machine learning to many different therapeutics and delivery methods. So here's 6 companies, thanks to my alumni postdocs. One is aimed at making antibodies, and you know how useful antibodies can be medically. Another one allows us to test. It's not just about a particular category of protein pharmaceuticals, but it allows us to test hundreds, maybe thousands soon, simultaneously in an expensive animal model, where you can, just like it with the previous one, which was delivery method, this is the payload itself, and it's got a protein barcode tag. Dino I already described for viral capsids. Jura is making using receptors on the surface of immune cells, using machine learning on those. Shape is on using RNA editing, using natural editors, not using foreign bacterial proteins, but using human proteins to do the RNA editing. And then Patch is on regulation, via engineering DNA and RNA with machine learning. So that's how we can capture machine learning for almost every aspect of everything I'm talking about here today. Now, the next few slides, I'll have lots of inspiration from zoology. Among other things, I'm a zoologist by training, and you'll see how important this can be in just a moment. So here is a database that includes humans as one of the animals. This is Maria, who is still walking around quite ably at 116. But these are outliers. These are all animals that are above the curve for when you do a mass versus maximum age curve. There tends to be a relationship, but the outliers are the interesting ones. And this comes from a human aging genome resource and an animal aging resource, both from Pedro de Magalhães, who was a postdoc in my lab and we're still collaborating many decades later. He maintains these wonderful databases that tell us about animal and human aging. So anyway, we're looking at these outliers, and even another set of even more extreme outliers. As you get closer and closer to human, it obviously becomes easier to transfer the know-how, but some of the most extreme values are far away from us. But the point of this is not we are harvesting some ideas for genes from very long-lived humans, near Barzillai and others have studied this, and these long-lived organisms, which Pedro and I and others have studied. But the critical thing that I want to get across here is the difference between the bowhead whale that lives 200 years and some rats that live 1 year is probably not their diet or exercise or yoga or metformin, some small molecule drug. Although those are all great, those are part of our solution for now for humans. What the difference is, they have fundamental genomic differences, and it may take fairly radical changes to reset. They may be some simple core principles, but it may take the ability to, we need to get our hands on the levers, and those levers are at the genome level. Not necessarily from birth. These may be things, as you'll see in a moment, these are levers that can be changed late in life. So where do we stand with this whole genome engineering? Now, we use artificial intelligence, as I already described, in a number of different settings, 6 different companies. But just like they can be used for DNA, RNA, and proteins, they can be used at the whole genome level. And we do that, and here are 2 killer apps. We can make organisms resistant to all viruses, as you'll see in a moment. We're trying to extend that to a variety of other organisms, including human, but we're not there yet. But I'll show you the one case where we have got it. We also can make our proteome, that is the collection of all our proteins, so that they are all essentially identical. We can make if we can program the genome radically, whole genome programming, either by multiplex editing or by a complete rewrite, then we can make the proteome, the collection of proteins, identical to a particular person. And we know that when you transplant organs between identical twins, you don't need immunosuppressants, which have their own issues. I mean, they're a real medical breakthrough that we need to celebrate, that allowed us to get to the point where so many people are receiving transplants, but they have their issues we'd like to avoid. Now, you've probably heard that we're not doing germline engineering on humans. The one person who did this spent 3 years in prison in China for it. No comment, but there are advantages, and we do use germline engineering, and it does make it into humans, but it's not germline engineering in humans, it's germline engineering on pigs, which we'll talk about in just a moment. But the advantages are delivery to all tissues, which is hard to do with conventional gene therapies on somatic, meaning adult tissues. It's a millionfold lower off-target risk. You can debug the cell whose nucleus will contribute to all the cells in the body via germline, meaning from a single fertilized egg to an adult. And via transplant to cell therapies, these are applicable to adults. Now, so that's whole genome design, germline. Somatic gene therapy is how we mostly deliver things, and those have their advantages too. Very short clinical trials. If you want to do germline, if hypothetically you could do some kind of germline in humans, it would take, and you wanted to say prevent a late onset disease like Alzheimer's, it would take you 70, 80 years to do the first round of testing. And if you needed to refine it, you'd have to go back to square one in another 70, 80 years. While if you're just trying to reverse the very early stages of a late onset neurodegeneration, then that's a shorter clinical trial. That's a normal length clinical trial. And also, another advantage of somatic or adult gene therapy is we're 8 billion of us are already born where we can't benefit from germline therapy. So if we could, and we're getting very close to being able to engineer the entire human or pig genome, what would we do? We wouldn't just solve the organ deficit, the shortage of organs, which is a tragedy. That's important to solve that, and we're working on it. But we would also make the organs enhanced. And by enhanced, that's just many of our technologies are enhancements. Our cell phones, our cognitive enhancements, our jet planes are enhancements for our means of transportation. So what would we do? And we don't want the organs or the cell therapies to fail for the same reason the original cells were failing. And so we want to deal with that. We might also want to have things that aid in the surgery, that make the surgery less expensive, higher quality, and so forth. Having to ship around an organ from a deceased individual in a timely fashion. It's very often that you'll have everybody all set up at both the donor surgery and thousands of miles away, the recipient, and then the organ arrives and the surgeon looks at it and says it's too old. It's too many hours past its prime. So anyway, one thing we can do that can help is to have the pigs ready. We could also have the organs, however they're made, stored. So cryopreservation and even dehydration are things that we can be inspired by the animal kingdom. For example, there's this Siberian salamander which can survive minus 55. This is very low temperature, and it can survive minus 35 for 45 days. So that's number 1 and 2 is cold and dehydration enhancements. But it gets more and more interesting as we go down the list from the 3, 4, 5, 6, and 7. So number 3 is DNA damage, which we know is involved in some of the aging components. There are many different aspects of aging we need to get simultaneously and thoroughly. I think it's not sufficient to get 1 or 2. This is going to be a classic case of polypharmacy, where you need multiple drugs. In any case, you may know that there are organisms that are exceptionally radiation or DNA damage resistant, but what you may not know is that it can be as few as 4 mutations that turns a very sensitive genome into a very resistant one, meaning 5 factors of 10, 100,000 times better with just 4 mutations. And again, we're trying to transfer this into humans from our experience in model organisms, so that we can enhance the organs that are being transplanted. So that's number 3. Number 4 is resistance to immune rejection. I have 2 examples of this. The first one here is the organ transplants that I've been alluding to. There's a number of very talented groups working on this. This particular thank you is to Luhan Yang, who was a graduate student and postdoc in my lab, who we co-founded eGenesis, and Jim Markman, who's one of the various surgeons that are doing the preclinical trials, where we now have up to 2-year survival of the organs. Now, to get these, we had incredible, and this is an idea that dates back to the 60s, where it was done with using chimpanzees, which is no longer ethical. And even back then, only 1 in 13 survived in only 9 months. So now, fast forward to today, they had the incredible rejection due to some of the sugars. There are 3 main sugars, so we had to knock out those genes. And we ended up, 69 genes had to be edited, various things involved in the blood, clotting and complement in the blood. And the MHC is short for the main thing that you match when you're matching two human donors. It's the same thing we need here. So, and then we made a lot of edits to eliminate certain pathogens, retroviruses that are built into the genome of pigs. This may not be necessary for every single case, but in the era of precision medicine, there may be people that will act as will be exceptionally sensitive to them, or there could be a chance mutation where you generate a zoonotic disease in a patient. These are things that just isn't worth the risk. It's much easier to do the editing. You may think that I'm making light of this, but we've done this in many strains of pigs, eliminated this viral threat. Now, second example, very different example. Here, we're not transplanting from pigs into primates, but from primates, humans, into mice. And here, the enhancement is making solving a autoimmune disease like multiple sclerosis that causes demyelination, loss of the myelin sheath that goes in the white matter of your brain that allows the neurotransmission to go exceptionally fast. When by putting in human differentiated cells we got from human stem cells, actually this experiment was done with cells from my body, that we can make both the neurons and the myelin sheaths, this wrapping around, and the synthetic version of this has exactly the same morphology and wrapping as the natural ones. And when it's put into a mouse, it restores it overcomes the autoimmune demyelinating, and it's enhanced because it has a particular communication via cytokines with the immune system that keeps it from being rejected. Now, number 5 is infectious disease. Infectious disease is a big part of aging. It is age-related. This is a age-related infection related deaths for a variety of different diseases. I have to inject the zoology here, which is there's several diverse, somewhat unrelated species of the vultures, rats, the Komodo dragon are examples. They're exceptionally resistant to infections. These can anthrax, plague, all sepsis, all sources of sepsis, and so forth. Part of this is due to their stomach pH, and a lot of it's due to their immune system. And furthermore, we should be aware that even the pigs, which are not quite in the category of these in their exceptional resistance, they are resistant to most human-specific diseases. A huge fraction of human diseases are human-specific: smallpox, polio, malaria, the list goes on. And the pigs are not sensitive to those. But they are sensitive to some pig-related diseases. It's unlikely that they're going to get a pig disease from a human when it's transplanted. So it's a net positive that they're resistant to human-specific diseases. But if we really want to eliminate a broader set, aside from being inspired by animals that are super resistant, we have come up with a synthetic biology approach that we think is very general, that allows us that we predicted back in 2008 that this would result in resistance to all viruses. It's hard to prove that it's all viruses. We can just say every virus we've tried, including random samples of viruses present in farm waste, sewage, and so forth. And the trick was a very simple one. There are 64 triplet codons. These are all possible combinations of A, C, G, and T that are the genetic code that go from DNA to RNA to protein. There's 64 of those. We took 2 of them, which encode the amino acid serine, and swapped them over to leucine. So every time there should be a serine, there's a leucine. Now the host cell, the good guys, were in on the game. We prepped them so they would not be affected by the serine to leucine swap. But the viruses throughout the world were not present in the genome modification process. And so every virus that comes in is every protein, every gene in every virus needs those two serine codons to be serine. They don't want them to be leucine. So they get swapped to leucine, which is chemically completely different from serine, or it's a mixture of leucine and serine, which is also bad. Anyway, we have not yet found a single virus of any sort, either laboratory viruses or wild viruses, that can break through this. And for sort of theoretical reasons, we think it's very unlikely that we will find any in nature. We could make them if we wanted to, but we don't want to. Okay, finally, number 6 and 7 in the set of 7 enhancements that we would like to make as we get better at engineering whole genomes. These are pathways for aging. In a way, everything I've been talking about is age-related. Aging affects almost every disease. But these are specific pathways for aging. We know quite a bit. I wouldn't say that we know everything, but we can't let perfect knowledge, perfect understanding be the prerequisite for engineering for health. I mean, it certainly didn't stop us for the smallpox vaccine back in the 1700s. We're using almost exactly the same protocols in the 1900s for vaccination. It's gotten a little bit different, as I just mentioned, is kind of now gene therapy, but we were right for 2 centuries, even though we didn't know anything about the immune system or virology. And we're in a much better shape now with the pathways of aging. This is showing the nucleus at the bottom and the signal transduction from the outside of the cell on the top, and the kind of the flow on the outside into the blood. And you've probably all heard that that there's important experiments on exchanging blood between old and young mice. We want to be able to leverage that for gene therapies, because until gene therapies can really deliver to every tissue with 100% efficiency, what we can do is deliver with slightly lower efficiency to cells that will then release it into the blood, and then the blood delivers it with additional efficiencies. And so anyway, if you look at, we're going to focus on three genes. We started with 45 gene therapies and tested them all out. It was actually fairly easy to do 45, thanks to Noah Davidson. And then took the 3 most promising ones that had the property of spreading through the blood. And here they are color-coded, red, blue, and yellow. Fibroblast growth factor 21, which Liz mentioned, Clotho, which she also mentioned, and TGF-beta receptor here. Now that doesn't normally float around, that stays in the membrane, but we made a soluble form of it. All of these were inspired by previous work in the literature. So they've been vetted partly either in germline in animals or other experiments that weren't gene therapies. We turned them into gene therapies, 45, which whittled down to 3, and then we did every possible combination. This Venn diagram shows those combinations. And here are the results of the gene therapies using initially 4 and eventually 7 different age-related diseases for testing. Not just biomarkers. Biomarkers are great, but we wanted to biomarker in principle like DNA methylation. You could get that right. You could get that perfectly right and still age. If you game the system, so you just change the methyl groups. Here we wanted to get at diseases and disease models that would convince the FDA. And here's one where you give them a high-fat diet to artificially induce the kind of obesity that can set in with age. And it reverses it, and it doesn't overshoot. It plateaus at the right age. You can see these magenta and blue lines for the particular combination of Clotho and FGF21. We did similar tests with a kidney obstruction, diabetes model, and heart. all of them showing promise with this small set of genes. Now, we don't go to the FDA promising longevity because that takes a longer clinical trial. The standard deviation for human longevity is 15 years, and that means the clinical trials are even longer. But every now and then, I mean, I showed you we're doing reversal of age-related diseases or models of that. But every now and then we'll let the mice live and see, just for curiosity to see what the longevity is. As long as we're curing diseases of aging, we can get the FDA approval. And if the side benefit is longevity, that's great. Here's a case that Noah Davidson and his coworkers published recently showing a significant blue line, significant post-treatment at the red arrow extension of life. But what's really interesting about that is not that significant improvement. It's when it was applied. This treatment was not applied when they were babies or children. This was applied at the point in the normal aging of the mouse colony, such that half of the mice were dying, perfectly healthy mice dying of old age. And nevertheless, you can see almost instantly longer life expectancy of these mice that are half dead. This is just another plug for Liz and BioViva. This is work that we published together showing the very similar but more dramatic shifts from the controls on the left to survival curves that are shifted to the right with TERT and follistatin, as she's already described. Now, Noah has participated in 3 of these 4 studies, all the ones involving AAV as the vector. Cytomegalovirus that we used with Dr. Zhu and BioViva is a much bigger vector. We haven't really fully utilized its size yet, but it's an interesting experiment. AAV is the one that is has the most FDA approvals and all of the improvements I was talking about with AI machine learning. So these are the 7 diseases that we've been testing so far in mice and dogs, and soon we'll be starting human clinical trials. Thanks to Rejuvenate Bio and Noah. And I just want to end on a particularly peculiar category of age-related problems. These are problems that have to do with the repetitive part of the genome. This is the dark matter. This is the part of the genome that made it such that we had not finished the human genome when we said we did back, not me, but my colleagues in 2001. And just now, this past year, we're getting to the point where we can almost deliver, we delivered a haploid genome, which is not quite the same as all of us that have 2 genomes in each of our cells, one from mom and one from dad. Anyway, these are the repeats, and they're not just annoying for sequencing. They actually are in all 4 categories are involved in various aspects of senescence, inflammation, and so forth. And here's some papers from a variety of laboratories showing this impact of centromeric and interspersed repeats, and so forth. And the telomeres we've already heard about is a 5th family. One of the companies I work with called Transposon Therapeutics is focusing on a small molecule drug that prevents some of these from jumping around, these jumping genes. But we wanted to, that is a partial effect. That's something that inhibits their jumping around. It doesn't completely prevent it. To prevent it, we wanted to use gene therapy. And this is where we set our record for gene therapy for the maximum number of edits. This is using a deaminase that changes A's to G's, where you target them with a CRISPR base editor. Thanks to David Liu, who provided these in advance of publication, and thanks to Raphael, Chunting, Corey, Oscar, Khalid, and Verena, who did the hard work getting this to work. So anyway, we've changed 24,000 edits in a single cell. Most people are happy, or if they can pull off 1 or 2 edits. This is where we're going towards changing the genome radically. 24,000 edits. And again, I just want to end by showing you something hopefully you've seen before, this hallmarks of aging. I've added in cancer here, so there's 10. Some people have 8, some have 12. But anyway, these are things that you all know about: caloric restriction, telomeres, senescent cells, and so forth, epigenetics. But I think we sometimes need to remind ourselves is we have to get them all right. It's whack-a-mole, where if you get one of these 10 right, you might get 2 years of longevity of life expectancy extension, hopefully healthy, youthful extension, but still you need to do more. You have to get all of them right probably to get more than 2 or 4 years. Period, full stop. I think we're in Q&A now.
Super. So great to have you with us remotely at RAADfest. So we're going to take some questions. We're going to do it live here. We got people passing mics, so you can just raise your hand if you have a question for Dr. Church. In the meantime, can you talk about targeting genes for organ regeneration versus functional age reversal?
So cells and organs are just another way of delivering genes, is one way of thinking about it. Delivering genes that either are going down with age, you can bump them back up, or you can put them in new configurations that are either genetically or epigenetically enhanced. So I don't make a big distinction between an organ transplant and a cell therapy and something that's rejuvenating and something that's regenerating. They're all part of a continuum that we need to get very good at engineering and replacing them. Some of the replacements will be minimally invasive, outpatient type operations. Others will require surgery, for example, heart and lungs, liver, kidney, and so forth, at least so far.
So it's all for the good. Okay, questions. Who's got a question for Dr. Church? Yeah, how about right here? Oh, wait for the microphone. So he's very far away.
Are you working on a gene therapy to correct the BRCA mutation that could avoid women having to have horrible surgeries?
Right. That's an excellent question. I heard it earlier today, and I wanted to answer it at that point. But the answer is twofold. One is, yes, we can address gene therapies to many things at once. So it could address it can address rare components and common ones at the same time, and then you benefit from the cost reduction of dealing with the common ones. So that's one possibility. I think the rare ones are going to be challenging, but there are things that we can do already that don't require any further FDA approval. For example, in vitro fertilization. Another one of my companies called Orchid Health does whole genome sequencing of every IVF embryo that goes through, and then they can select the ones that don't have the BRCA mutation. So that's an example of something that one can do today, building up the next generation. But just like what I said about why we don't do germline engineering, or why it's hard to get FDA approval, is that many of us are already alive, and those who are alive with BRCA1 will need either gene therapy or preventative surgery.
Okay, can we get, here's some exercise for somebody. Oh.
I'll hold the mic.
Oh, thanks. How can a motivated, somewhat talented person help volunteer to advance this technology?
Great question. We have genomics projects where we can learn what the correlations are between people who live a long time, for example. And some of these are done at a national scale, like all of us in the United States. So the UK Biobank, there's efforts in Iceland and United Arab Emirates and so forth. So that's one way you can just get involved in these genome-wide association studies. Personal Genome Projects was one of the ones that I helped start. The other one is spread the word, get educated about the details. This is really not that much harder science than what's going on in your cell phone. Advocate, raise money, et cetera. There's many things that can be done.
Do you have
Get your genome sequenced. There you go. There's something you can do. It's only $250.
Do you have a longevity escape velocity sort of projection where the idea is that you're reducing life expectancy? I mean, you're adding life expectancy faster than offloading it. Do you have one of those?
Exponentials are wonderful. It's nice to almost my whole career I've been dealing with exponential technologies. It's terrific. You can look good just because it's exponential, but it's also hard to predict. But typically it's hard to predict in the unexpected way. The things arrive ahead of schedule. So for example, the Moore's Law curve, which was a pretty good curve, predicted it would take 6 decades to bring down the cost of genome sequencing from $3 billion. Instead, it took somewhere between 6 and 10 years. So the escape velocity is something where we've been poking along for about adding a 1 year every 4 years, which is not nearly good enough. We've been doing that for about 170 years. It's getting, hence we live about twice as long. Most of those, there's some, but with these new exponentials of biotechnology kicking in, we may get our first year where we get more than a quarter of a year of advance, and then so I don't know when that would be. I wouldn't be surprised at the rate we're going. It would be within the lifetime of many of the people in this room.
That's good news.
We get to that point, that break-even point.
Yep. Can we get
Hi there. I'm curious more about the transposons and the dark matter, you say. Do you see a stage where we're going to want to go in and get all of the transposons out of the human genome? Or is that something we're just going to let them be and we're okay?
Well, there's an interesting story. I'll try to keep it short. But the transposon therapeutics company is using drugs which essentially neutralizes their hopping around without removing them. So that's indicating where we might be going. As we get the ability to engineer the whole genome, we may not want to remove them. We may want to mutate them so they don't hop around. That seems to be the main thing that's the problem. And mutating them so they don't hop around is 100%, while the drugs are probably 90%. Now, 90% may be adequate. We're going to find out when we go into clinical trials. But just, it's plan for the worst and hope for the best. Our backup plan is to edit them. And the reason we might not edit the whole thing is illustrated back to the pigs, where we had similar hopping elements, virus-like elements that would jump out and infect or hop around in the genome. In that case, we did not remove them, although we could have. It's a small number. It was 25 initially. The reason being that that one of the genes is actually useful. The envelope gene. This is like the spike protein in coronavirus. There's an envelope gene on the outside of retroviruses, and that has been co-opted by most mammals. It is part of the placental development, and so it's actually essential for them to have healthy baby pigs. So we left that intact, and it had the side benefit, it had a second function, which is it protected it from getting reinfected by the virus that we had just eliminated, because the surrogate mothers, had not yet been engineered, and they were just full of these viruses. And we've shown this over and over again in multiple strains that they do not get reinfected because probably this envelope protein is protecting it. So it has two functions: protects against viral infection, and it leads to the healthy development of the placenta.
Can we get this gentleman in? Yeah. No. Well. Yeah. Him. Yeah.
Though this may not be a zero-sum game, have you considered the impact of EMF with all the gene altering and then the satellites and relative to 5G and the impact it might have on the work that you're doing?
Right. There are actually fewer and fewer zero-sum games. We're getting into an era of more and more win-wins. And I think that EMF is much more subtle than all the things that I've been talking about so far, but it doesn't mean that it's out of something that it doesn't mean there's something we can't have a biotechnological solution for. Right now, I'd say it's early days, and fortunately, it's not the kind of killer that heart disease and cancer are. But yeah, I think it's a great question. It will be addressed kind of in order of public health threat level. It's my guess.
Okay, time for one more question. Let's have Jose here.
Congratulations, Dr. Church. I love all your work, all the things you do. And when I met you at Singularity University like 15 years ago, you talked about a totally different idea, which was de-extinction. You were working on bringing back the mammoth. What is the latest on that?
Thank you. You can think of de-extinction as the ultimate in de-aging. And the latest is we just in 2021 just got decent funding on that, very, very decent funding, and it's going very well. We have at Colossal is the startup company. It's now up to 114 employees, plus 2 major universities, one in Melbourne, Australia, under Andrew Pask, and in my lab in Boston. And we're going to be publishing a bunch of papers at the end of this year on multiple species and multiple new techniques. So we're already having some of the techniques we're developing. It's mostly about techniques that they're relevant to human health as well. So, for example, we have some that are being used in IVF clinics to mature the immature eggs, but via granulosa cells that we've developed from human stem cells. We have this multiplex editing is useful. Both, it's kind of a 2-for-1. It's useful both for bringing back extinct genes, but it's also useful for the pigs and so forth. So it's all been very much de-risked by the pig experiments. Making 69 changes in pigs is probably very similar to the process that we are using to make a similar number of mutations in elephants. So that's the short version of the update. It's looking very, very good.
Super. Well, thank you so much, Dr. Church, for joining us at RAADfest. Great having you with us. Thank you.
It's great being part of this community. Thank you all.